Thank you, Anita. Hello, everybody, and you're very welcome to this evening's webinar. My name is Humphrey Jones. I'm the Head of Science at St. Columba's College in Dublin, and I teach biology, junior science. I'm also a guidance counsellor, and I'm the current chair of the ISTA as well, the Irish Science Teacher Association. And I'm co-author of Evolution with a good friend, Yvonne Higgins-Vallely, who's Head of Science in a college in Bundoran in County Donegal, also a biology teacher, chemistry and maths, and a former chairperson of the ISTA. I've known Yvonne for over 20 years, and it's been brilliant to work with her professionally on this project over the last number of years. So the plan for our webinar this evening is to go through each of the following points. So we'll talk a little bit about the structure of the specification, how it's laid out. We'll talk about building units of learning while studying the specification. Working with the learning outcomes within the specification, and particularly around outcomes which deal with modelling. We'll talk a little bit about cross-cutting themes and how we think it's best to incorporate that into our teaching. We'll talk about, of course, being a science, managing practical investigations, and how we can build the lab skills amongst our students. And of course, with the new specification, we have to talk about planning for assessment, with an eye on the Leaving Certificate exam, and of course as well the new additional assessment component, the biology and practice investigation, which the students will complete over the course of their studies. So maybe just to start off talking a little bit about the structure of the specification. You've probably all seen this diagram, excuse me, this diagram here, which is kind of summarizes the whole specification really, and you can see here that there are 4 strands of the course. The unifying strand, which is the nature of science strand, which is at the core of the specification, and then 3 additional strands: organization of life, structures and process of life, and interactions of life. Around that then, there are 3 cross-cutting themes: sustainability, health, and technology, which blend into all of the various different parts of the strand. We'll have a little look at those strands in a bit more detail in a second as well. It's also important to kind of keep in mind the key competencies for a senior cycle, because we know that the students can be assessed on the content of the course, but they can also be assessed on these key competencies as well. So these are the kind of things we're trying to develop within our students over the course of the 2 years. So whenever we talk about the structure of the specification, the most common question we get is, well, what's new and what's gone? So here's a little slide on what's new and what's gone. So what's new are elements called phylogenetic classification, so a little bit of extra detail on classification. Epigenetics, obviously relates to genetics, a little bit more information on that. Bioinformatics, a really interesting concept that I think we'll really enjoy teaching actually, and getting our students to look at and analyze genome databases. There's a little bit more information on artificial selection when we're teaching evolution. And I think a really nice addition to the reproduction section is prenatal and postnatal care, which is really practical advice and practical content for our students. Within ecology, there's a couple of extra pieces as well. Species diversity index is something that I think you probably explored on your IJED training day already. And also the concept of carrying capacity has been introduced into ecology as well. Probably the biggest change really is looking at how the cross-cutting themes integrate within the course right the way through. So we'll talk a bit more about those a bit later on, but there's something that we always have to have in the back of our minds as we're teaching any particular topic as well. And of course, just like we have with junior cycle science, there's now a much greater emphasis on the nature of science, the skills associated with being a scientist. So what's gone? Well, the main topics which have been taken away are the sense organs, so the eye, the ear, and the skin. You don't have to teach it all. And the lymphatic system, which is, we don't have to teach as a unit, although we probably will reference it from time to time, especially when we're talking about, say, digestion or circulation, etc. as well. Now, there are other things which have gone as well, but they're really more simplifications of things that we're maybe already comfortable enough teaching. So, for example, plant reproduction is a little bit thinning out of some of the content we have to teach there, but probably it's not worth mentioning everything which has been thinned out slightly in relation to this. But you can see, though, overall, though, that there's definitely more added to the specification than has been taken away. And that will have its own challenges we'll talk a little bit about later on as well. So let's look at some of the strands in a bit more detail. So as I said, within the nature of science strand, there are 3 subdivisions: investigating in science, science and society, and scientific knowledge. Now, they're common with the chemistry and physics courses as well. And then we have our own little part of the nature of science strand called biological reasoning, which obviously then reflects on how to think like a biologist and how to learn like a biologist. The first core strand is called organization of life, excuse me, water. And as you can see, there's kind of 5 areas and 5 divisions within that. So the characteristics of life, probably all very familiar with teaching. The chemicals of life, looking at biomolecules. The units of life, looking at cells. The origins of life and evolution, including, as I said earlier on, a little bit on artificial selection. And then information of life and looking at genetic inheritance. That's all within the organization of life strand. Then we have the second strand, the largest of the strands, structures and processes of life. And this looks at things like enzymes, cellular processes like photosynthesis and respiration, transport and transfer, again, so looking at physiological processes like, say, for example, transporting plants, transporting humans, reproduction, both plants and humans and animals, response, so again, plant responses and animal responses, and the information of life as well, cell division and protein synthesis. Now you notice that the information of life has cropped up again as well here. And then the third strand is called interactions of life, and this is the shortest of the strands. So it includes mainly ecology, ecosystems and biodiversity, microorganisms and nutrient recycling, which often go with ecology anyway. And again, we have information of life here within and looking at genetic engineering. So in a lot of ways, you might think that information of life probably could have been a 4th strand by itself, but I think they wanted to emphasize the interconnected nature of the course. Now, interestingly, both neither the NCCA or IDA recommend teaching the course strand by strand. So the course isn't laid out in the order that they want us to teach it. And this is very much in line with how they structured the agricultural science specification when it was introduced. And what they want teachers to do then is to teach across the strands by building a series of units of learning and what we call the thematic approach, or what they call the thematic approach. And so they're challenging teachers in order to build units of learning. So let's talk about that as the first aspect of what we're talking about tonight. So as I said, IDA are recommending using this thematic approach, so not teaching strand by strand. So what this means basically is we take a particular theme, say for example, blood, and we see which learning outcomes in each of the strands reflect or might link in with that. So obviously you might be looking at things like cells, you're looking at health and disease, looking at transport and transfer, all of these various things. And you build a unit of learning, dragging learning outcomes from each of the various different strands. Now, I think it's really important to note that, you know, as a teacher myself and as somebody who went through, in particular with ag science, I knew this was kind of what students or what the NCCA would have liked us to do. So when we set about writing evolution, we straight away set up a structure whereby we would divide the book, not in the strands, but according to the themes. So we have created 12 broad units of learning, thematic units of learning, incorporating learning outcomes from across the strands, and then of course including the nature strand, nature of science strand as well. So we have kind of structured the book in a way that we've broken down into the various units. Those units are broken down naturally into chapters, but we've actually gone further and broken down those chapters into individual sections, effectively lessons for teachers to aid with their planning. And we're all probably having nightmares still about being asked to unpack learning outcomes at junior cycle science. Well, we've done all that unpacking for you, and that's built into the sections as well. So you can see here within our table of contents, for example, there's our 12 thematic units of learning, and we build the chapters around that as well. Now, we'll all want to be updating our schemes of work in relation to this, of course, and one of the aspects of evolution that we really think will help support teachers as we transition to the new course, including ourselves, of course, it's very, very useful, is we've got a really comprehensive teacher resource book. And on that teacher resource book, we have put in an editable, so a scheme of work, which is using that thematic approach. So breaking down into the various units. You can see here a sample of the unit of learning for the first unit, the living science, introduction to biology. And you can see how we've broken it down then, incorporating the learning outcomes, the key competencies, the practical activities and assessments as well in relation to that, and the cross-cutting themes. So that information, that I want to assure that aspect of your planning, where you might have to take you plan your units of learning, we've done it for you, built into the book directly within it as well. So then, I suppose the second biggest challenge, and again, probably reflecting on junior cycle science again, where there maybe was a bit of ambiguity around understanding the detail required when reviewing a learning outcome. We wanted to make sure that, again, that when writing the evolution, that we really wanted to make sure that a lot of that ambiguity is taken out for the teacher. So we spent a lot of time unpacking these learning outcomes as well. So we know they're difficult to unpack. They were difficult to unpack for junior cycle science. They were difficult to unpack in agricultural science as well. But I will say that the new specification biology has made improvements, and they now, instead of just using learning outcomes, they now use a hybrid model. I'll show you a little stretch of the or a little cutout from the specification in a second, just so you can kind of see this in practice. But there's far more detail now in terms of understanding exactly the depth we need to teach as well. So now I will say that it's probably not perfect yet. So there's still a little bit of ambiguity. Still sometimes you're scratching your head wondering, what do they mean here? What kind of depth do I have to go in here? But Yvonne and I spent a huge amount of time unpacking all these learning outcomes to make sure that we knew exactly what needed to be put into the textbook, so you don't have to spend that time unpacking those learning outcomes. And I'll show you how we've actually incorporated that directly into the book as well. So when you're looking at the learning outcomes, and it's probably for yourself, but I said we've probably all this work done for you anyway. When you look at a learning outcome, what they're asking you to, first of all, focus on the action verb that's in the learning outcome. So for example, if a learning outcome begins with the word outline, then you're just giving the main points. You can see it there, giving the main points, restricting to essentials. While if you're asked to describe, be asked to provide an awful lot more detail in relation to that. Interestingly there, you can see that word model on the very top of the screen there. I'll come back to that in a little bit moment, because there's quite a lot of learning outcomes in relation to the word model, and I know that it's caused a little bit of anxiety for people, so we'll talk a little bit about that in a moment. So I'm sure you've had a chance to look at the specification at some stage anyway, maybe not to study it completely, but the best way to look at the specification is actually to start on the right-hand side in the grey boxes with the learning outcomes. And you can see there, outline the cell cycle. So straight away, this tells me I don't need to go into that huge amount of detail on the cell cycle. I'm restricting to basics. And then the parts on the left indicate what aspects of the cell cycle the students need to know about. So at ordinary level, they need to know about growth, DNA synthesis, and division. While at higher level then, which are indicated in bold, they need to know a bit more information about interphase, and need to know about nuclear division and the 4 stages of mitosis. They need to look at cytokinesis and how that process occurs. And again, the concepts of diploid. Likewise, you can see the second one, compare the roles of mitosis and meiosis in transmitting genetic information. And then the context, the depth that we have to teach that is on the left-hand side as well. So you can see not a huge amount in terms of the comparison there. Genetic information remains the same, variation of genetic information in meiosis. Now, the learning outcomes that begin with the word investigate relate specifically to scientific investigation. So experimental investigation that students have to carry out themselves. And in particular, be mindful of investigate where it says use primary data. So in this case, using primary data gathered with a light microscope and secondary data gathered with scanning electron microscope images. So the students are expected to gather that data themselves by using the light microscope. And again, you can see on the left-hand side, the context that's provided there. So again, microscopy, the idea of microscopy, preparing and mounting a slide for viewing in the light microscope, then students must be able to do that. And then they need to be able to compare all those parts that are labeled there. You can see there in higher level, a couple of new ones compared to the course that we're teaching at the moment. We now reference endoplasmic reticulum and Golgi apparatus. So what we've done with the learning outcomes, as I said, is we've tried to unpack these for you. And we have structured we've broken the strands up into our units of learning. We've broken those units of learning into chapters, and then every chapter we've broken down into sections. And each section is designed to be, I suppose, a class, a lesson. And you can see here, each lesson then, we say, right, this lesson here, section 1 in cell continuity, there are learning outcomes that we've unpacked from the learning outcomes and specification listed below that. And we've been able to take it a little bit further because we broke it down into the sectional structure. We were able to provide, at the end of each section, a little portfolio link. So within our learning portfolio, which is a part of the 2-in-1 workbook that students get when they purchase Evolution, there is an assessment task directly linked to every single section. So for a teacher, like myself, as I'm going to be using this, I'm looking forward to using this, is that when you're going through a section within the class, you have a ready-made homework task directly afterwards, and the tasks are designed to be carried out quickly. They're low stakes. They're checking for understanding, but you're checking their understanding, and they're checking their own understanding as well. So to give you an example, again, so here's one on mitosis, a detailed study. You can see here we've unpacked the learning outcome to see, well, what does that learning outcome mean? You can see 2.3.1, and we've written out, these are what we think they're in red because they're in higher level. And again, we've got a little portfolio task, as you can see there. So when you finish that task, direct the students to the learning portfolio, and they've got a task, really straightforward, easy, quickly achieved task that you can quickly check their understanding, and they can check their own understanding as well. So it's formative assessment built into the textbook, which I think is quite a unique take. And again, you can see here, there's the third section. Again, in this case, it's related to cancer cause and treatment. There's our lesson, there's my learning outcomes, and then I have my portfolio task directly built into it as well. So we think that's a very useful structure within the textbook. It's not just providing information, it's actually providing an opportunity for students to check their understanding as well. Just mentioned very quickly in relation to modeling, there are 20 learning outcomes which begin with the word model. And a lot of teachers might be a little bit scared of this, but actually I think it's really important to note that we're always modeling, that teachers are always modeling, good techniques, we're drawing good diagrams, we're showing students how to use equations. And you can see there, according to the specification itself, modeling uses words, diagrams, equations, physical models, or simulations to represent ideas, structures, and processes. So it's not just about actually building or using physical models to describe how things work within it. I think it's really, really important to note. I think that it's a very common misconception, I think, initially, and even heard it out there amongst various training days as well. So we use models. So if we're, for example, describing mitosis, we're going to draw diagrams of each of the stages. That's modeling. So we use models to help students understand how to use models. So we draw the diagrams, we go through, we teach the students how to draw the diagrams, so then learn to use models as well. And there's a lovely quote about modeling. It's, the mediocre teacher tells, the good teacher explains, and the superior teacher demonstrates. And for me, modeling, when I think of modeling, I think of the I do, we do, you do it together, you do it alone structure, which is, say, for example, I'm trying to teach students a particular lab skill. I will demonstrate it, I will show it. The students will repeat it all together. Then they go off and do it in groups, and then they get a chance to do it by themselves. So I might model the microscope, then we do it all together, we repeat it. They go off and do it in groups, helping each other out, and then they do it alone. So it's something we do all of the time. It could be the same with a maths problem. We do it together, off you go and do it in pairs, and then you do it by yourself as well. So I think the most important thing, hopefully, don't worry about that idea of modeling. I think it's something that comes naturally to science teachers, and I think it's something that we're going to naturally link in with anyway. So moving away from the learning outcomes, and as I said, hopefully, we don't even have to look at the learning outcomes and specification. Hopefully, that knowing that that's all been unpacked for you. But working with cross-cutting themes is an interesting area as well. And again, this is following a little bit from ag science, which has quite a lot of cross-cutting themes. But biology, chemistry, and physics collectively have 3: technology, health, and sustainability. They're shared across the 3 senior sciences. And what this is for a way for us to view each of the topics that we're teaching, to make sure that we challenge the students to see relevance in what we're teaching in their own daily lives. So say, for example, you're teaching about the heart, for example, the heartbeat, maybe you're referencing things like technology. So for example, maybe things like heart rate monitors. And then that relates obviously to health in terms of that too. And you could even bring in sustainability there, because we've got maybe the plastics that have been used to build it, for example. You could have a discussion around that as well. So And I think it's really, I think it's important to think that it can be a challenge for teachers to find relevant content that might relate to individual topics as well. So what we've done within Evolution is try to, in every single chapter, provide a case study, maybe 2 or sometimes even 3, on one or more of the cross-cutting themes into the chapters. And we also start every chapter with a little eye-opener, a little story to kind of get students discussing it. And again, they often relate as well to one of the cross-cutting themes as well. So you can see here, for example, there's a cross-cutting theme in relation to sustainability, and we're talking about viruses, which is quite interesting. Then you see one here on health in relation to natural killer cells, so our immune system. Obviously very easy to find a link to health and the immune system. And then for science in society, you can say technology, so even just ecology, using their phones to help identify identify plants in the ecosystem or animals, or maybe even to measure abiotic factors as well. So cross-cutting themes, I think they're not something to be afraid of. I think they're actually enhance what we're learning, and we do this a lot anyway. And it's just making sure that the students are aware that we're trying to find meaning. And so we give those case studies, but we can allow for students to incorporate further ones as well. In terms of managing practical investigations, the most common question I've got so far is, are there mandatory practicals? And the answer is not very clear. It's yes and no. There's no official list of mandatory investigations, and I think they were very keen to not provide that term. But what they have done is they provide 13 learning outcomes with the word or the action verb investigate in it, or certainly investigate using primary data. So this is that the students have to investigate and they have to gather their own primary data. So what we've called them within the textbook are specified investigations, and there's a mixture of short ones and more complex investigations as they go along there as well. Now there's obviously scope for a lot more than 13 investigations as well. So within our teacher resource book, we provide at least 2 suggested practicals for each chapter as well. So other experiments the students can do, but they're also going to be exposed to maybe secondary data, which can still incorporate those skills and develop those skills as well. Now, some learning outcomes, investigative learning outcomes, require students to investigate multiple factors. They might have to repeat an experiment more than once by changing different factors as well. And of course, students are required to keep a record of their investigation results. So I'll give you an example of one of the learning outcomes that relates to this. So here's one: investigate the factors affecting the rate of transpiration in plants. Use primary and secondary data to support your conclusions. And you can see on the left-hand side then, it lists the factors. You can see air movement, temperature, surface area, presence or absence of cuticle, light intensity, and CO2 concentration. So what in this scenario, the students are actually expected to be able to and understand how to gather primary data for all 6 of the factors. That's an important point, actually. Now, they might not gather the primary data for the 6 factors themselves. Maybe you've got a class of 24 and the students are carrying out the experiment, and you have 6 groups, and each group is doing one factor. So the students are gathering their own primary data from one factor, and they're gathering secondary data from the other students in the class as well. That's an interesting one, and I think but I think it's really important to know that students won't have to gather the primary data themselves, but they will need to know how to gather the primary data for each of the various aspects of the experiments. Here's another one as well: investigate factors affecting the rate of photosynthesis. So again, we might be relatively comfortable with measuring or investigating the factors affecting rate of photosynthesis, like looking at light intensity and CO2, but temperature now also comes into it too. So again, the students will need to know how to do each of the 3, but they might only gather primary data themselves on one of them. Now, of course, any good textbook will try to incorporate these seamlessly into the textbook itself. What we've tried to do, again, trying to be a bit unique and kind of making sure that it appeals to all kinds of learners, is provide a visual cue where possible for each of the investigations. So you can see here's one of our specified investigations, in looking at the organelles of plant cells and animal cells. And not only we have got a really clear method, but we also then have a very simple illustration to show the students how to do each of the various different steps as well. And as I said, the students must keep a record of their investigations. So within within our 2-in-1 investigative logs, or workbook, which is 1/2 investigative log, 1/2 a learning portfolio. They have a space to not only record their experiments, you see here one for chapter 18, but also there's questions basically on that as well, which again might mirror the style of questions that come up in the Leaving Certificate as well. Certainly what we anticipate them to be too. And so one other aspect we've done, because obviously we're conscious of the book being important, but in order to provide other opportunities for learners, we've created a series of experiment videos. And so I'm going to show you a sample of our video, which is me, very handsome devil over there, showing one of the videos of the experiments. And we're going to play this for about 5 minutes. Just get a break from my voice, you'll hear my voice. But hopefully you'll find it useful as well, give you a sense. And they're designed to provide an introduction for the experiment for the student, or as a revision tool for the teacher as well. Here we go.
Hello everyone. In this video we're going to explore the fascinating world of cells by preparing and observing both animal and plant cells under the microscope. For this investigation we'll need a microscope, iodine solution, methylene blue, some glass slides, some glass cover slips, a mounting needle, forceps, a paintbrush, some clean cotton buds, and a dropper with some water. First, let's prepare our cheek cells. To do so, gently scrape the inside of your cheek with a clean cotton bud. Add a drop of water to a clean glass slide and gently rub your cells onto the glass slide. Add 1 drop of methylene blue solution and then, taking a clean cover slip, gently lower it using a mounting needle at a 45-degree angle. To ensure we don't get any air bubbles. Use some tissue paper if you need to remove any excess stain from the slide. Now we're going to view this under the microscope. So next, place our slide on our microscope and secure with some clips. Select the lowest power on the microscope and adjust with the coarse focus wheel. Once you get a clear image of your cells, you can now increase the magnification and adjust with the fine focus wheel.
Okay, so if you want to see the rest of that video, you can. That's available on the Gill Education website, where you'll find loads of other digital resources as well for you to explore. We'll talk a little bit more about those towards the end as well. Okay, let's talk about planning for assessments. We've talked about, I suppose, planning our scheme of work, planning our lessons, planning our units of learning, and hopefully a lot of that planning has been for you. But obviously, planning for assessment is also a major part of any introduction to a new course. And within this, we have to think about 2 aspects. Of course, we have to talk about formative assessment. So as teachers checking our students' understanding. And as we mentioned already, that's built into our learning portfolio. So each of those tasks is directly linked to the end of every one of those sections. So it should be really easy for us to track and for us to check for student understanding for the pupils and for ourselves as well. But in terms of summative assessment, is we're looking at tracking learner progress, and of course the final examinations as well. And of course, both of those are good features of any well-designed textbook. So as I mentioned before, we've got a learning portfolio which has all those various different tasks, but we also have then at the end of each chapter within the textbook a series of end of chapter assessments as well. So just to maybe quickly talk about the learning portfolio, you can see another little learning check there in relation to that. We've also tried to bring variety in there. So by looking at the agricultural science examination papers over the last number of years, and also junior science as well, we kind of get a feel for the style of questions we're expecting within the Leaving Cert. I don't think they'll strain too far away from the model they're using for Ag Science. And in that, then they've got a wide variety of questions. So like fill in the blanks or labeling diagrams, or also maybe reading comprehension. So again, we've tried to incorporate some of those reading comprehension aspects into the learning portfolio as well. And then for our end of chapter assessment, try to do things a little bit different. We wanted to make sure that we cater for all style of questions, low order questions, data analysis questions, and higher order questions. So we've structured our end of chapter assessment questions based around Bloom's taxonomy. So we have students where they're asked to remember, understand, and again, analyze, evaluate, create. So a real variety of questions there. And I think in particular, we wanted to focus a lot on the analyze. So we provided lots of opportunities for students to look at data, to analyze data within the analyze sections of these questions, either tables or graphs, and try to extrapolate meaning from those as well. Now, a big aspect of the assessment, of course, as well as the exam, and we think that the style of questions in both the end of chapter assessment and the learning portfolio will help us incorporate that. I should have mentioned also within our learning portfolio, we also will put in topic by topic the sample paper questions as well that will come out. We're expected to come out in April. They'll also be built into the learning portfolio as well. So we will see exam sample paper questions in there as well. But the exam is only a part of our assessment, of course, for the new course. We also have to be mindful of our biology and practice investigation. And this is this new additional assessment component, which is being introduced into Leaving Cert biology, chemistry, and physics, of course, as well. So what do we know about it already? Well, we know that the State Examinations Commission will set a brief sometime around January of fifth year. They will provide a sample brief from September, so we have an idea of what to expect. But the actual brief that the students will get will be in January, likely of fifth year, sorry, my apologies. And then over the course of a year, but using 20 hours of class time, the students carry out all the various aspects of the brief. So they will carry out scientific research on the topic of their within the brief, experiment, design their own experiment, gather primary data, analyze that data, form conclusions based around that, and of course complete our report. I think we've got a lot of lessons again to learn here from agricultural science and their individual investigative study. And also the NCCA have issued a series of guidelines as well. And we've tried to break that down a little bit within the textbook as well to make it a bit more student friendly. Those guidelines very much were aimed at teachers, and we try to make it a bit more student friendly as well. So there's two aspects of our book which we're trying to focus on in relation to the biology practice investigation. One, that dedicated chapter at the end of the book, which focuses directly on that. I'll show you a little screenshot in a moment. And also within the investigative log, there's a dedicated space as well for students to keep track of their ideas, jot down their initial experimental procedures, maybe adapt them and reflect, basically log and reflect on the whole process as they go through it as well. So I think they'll be a really good addition for that too. Here's a couple of little screenshots from our chapter on the biology and practice investigation. You kind of see it gives you a little bit of a breakdown in terms of what to expect, written in student-friendly language, and also providing tips for success as well. So I think we've tried to incorporate that very strongly within the package as well. So we're coming towards the end of our webinar, and over the last 35 minutes, we've looked at the structure of the specification, how we can use that to build units of learning, how we can work with learning outcomes, and in particular with modeling, how we can plan to work with cross-cutting teams and incorporate that into our lessons right the way through, how we can ensure that students are developing their practical skills by carrying out practical investigations, and again, as teachers, how we can plan for assessment and plan for that biology and practice investigation as well. And I hope, and you know, myself and Yvonne have worked so hard on evolution over the last 5 years, that we think that we have ticked all of these boxes. We think that we have created a unique textbook that takes a lot of the planning out of the teacher's hands, because we've built those units of learning already. We've unpacked those learning outcomes. We've incorporated those cross-cutting themes within the planning. We've provided schemes of work within our teaching resource book That's editable, it's downloadable and editable from the Gill website as well, from Gill Explore, so you can adapt that to your own needs as well. And we think we've incorporated by providing really step-by-step and visual guides for each of the practical investigations, specified investigations within the textbook, but also within the teacher resource book too as well. And finally, within relation to planning for assessment, I think our package really provides something different for assessment, because it focuses on not just summative assessment, but formative assessment as well. And again, we're all a bit nervous, probably the biology practice investigation, but I think I think it'll something that we'll probably take a little bit of time to transition to, but I think hopefully with the right supports around us, including what we have provided in the textbook, we will sail through it over the next number of years. Thank you all very, very much for listening today. We're not finished yet. I'm going to invite Anita to come back in, and she's going to, and Yvonne, and we're going to take some of your questions as well. Thank you very much again.
Thank you so much, Humphrey, for a great presentation. Lots of really helpful and practical support there for Leaving Cert biology teachers, I think, both around specification and around how you tackled it in evolution. So well done. Yvonne, welcome very much. You're welcome. Sorry, thank you for joining us. Yeah, great to see you. Okay, so we are going to go into the questions. We had quite a lot that were submitted during the registration process, and a few more have come in now during the presentation. Humphrey, I think, has addressed a lot of your questions during the presentation, but we will just go through. As I say, I'm really keeping an eye on the time as well this evening, so we don't keep you longer than necessary. So we're just going to tackle, I'm going to take the first one because it's more from me. So Liz has asked, can I get a sample of the book, please, and access to online resources? So again, I think Humphrey kind of mentioned this, Liz, but just to say that currently on gilleducation.ie, there's a sample unit online there now, and there's also samples of the digital resources that Humphrey referred to, including that experiment that the investigation that he showed you a piece of. The full online book, so the complete book, will be available at the start of next week. So you can pop onto Gill Education and have a look at that then, and you'll be able to see all of the features that Humphrey has gone through and told you about this evening. And then the actual book itself is going to be going out to schools the week after Easter. So when you come back from your Easter holidays, it should be hitting the couple of days after that. So give it a chance to arrive. If, say, by the middle of that week you haven't received it, please do get in touch with your rep. They will be able to sort you out and help you. But do check out gilleducation.ie at the moment, because, as I say, the full book will be up there at the beginning of next week, and the sample resources are there currently. Okay, so we had Colette asking us what resources will be available with the book, PowerPoints, solutions, experiment videos, etc. Again, you've kind of covered that, but I don't know, do you want to say a bit more around that?
Yeah, I can take that. Thanks, Anita. So in Evolution, Humphrey and I, we've really built up a very comprehensive package that will support us as teachers and all other teachers out there. We're working in the classroom every day, so I think we know what teachers need and what's going to really help them on the ground come September. So in our teacher resource book, we have a very comprehensive scheme of work for both 5th and 6th year, as Humphrey has mentioned. One thing that I will be absolutely delighted to have come September is that we have all the solutions to every single question in the textbook and every single question in the learning portfolio. All the solutions for those questions will be provided. So I think that's going to be really invaluable to any teacher, including myself. Also, as Humphrey mentioned earlier on, we have additional investigations included on our scheme of work, because those that are specified in the syllabus, that's a minimum requirement. So we have at least 2 additional investigations mentioned in the scheme of work. And also for the specified investigations, we have background information for the teachers to assist them in planning and preparing and helping their students in the classroom. And I know myself as a teacher, I find that really, really useful, that kind of additional information. with regards to their digital package, as Humphrey mentioned, we have our editable PowerPoints. We have quizzes that will provide instant feedback to our students, so that's great for them taking control of their own learning as well. And obviously then we have our experiment videos, and I love to use them myself if I'm introducing an investigation, or sometimes afterwards as a recap as well. So I know myself as a teacher, I'm really looking forward to using evolution with my own students, and we've put a lot of thought, myself and Humphrey, into what's really going to support teachers in their classrooms in September.
Brilliant. Thank you. Comprehensive answer, Yvonne. Thank you. And just double checking, the solutions that you mentioned are all in the Teacher's Resource Book. Is that correct?
That's right.
They'll
That's right, yeah.
Great. Okay, just to clarify that. Brilliant. Great. So Maureen asks, do you think the methodologies and approaches to teaching will be radically different with this new course?
Yeah, thanks, Anita. Absolutely. I think as a teacher, all you need to do is look at page 41 of the specification, where they have outlined the approach teachers are going to require for learning, helping their students to learn the new specification. And discussions is going to be, I suppose, a big change in the classroom come September. Students are expected to take part in group discussions. So we've really built that into evolution. You'll see throughout the chapters, there's think, pair, share. And in our end of chapter questions as well, we have opportunities for debates there as well. As I've mentioned, the investigations that are specified in the course, that's a minimum requirement. So we have also all our additional investigations. So teachers will have to do extra practical work with their students. But I think one of the biggest changes is the cross-cutting themes, and how that really is permeating the whole specification. So as you saw earlier on with Humphrey, we have our cross-cutting theme boxes, and again, they're going to be spurs for a discussion in the classroom. So it'll really help support students in developing their critical analysis skills. And finally, I suppose students have to be able to interrogate and interpret data, and we've built that in both in our learning portfolio and in our analysis section of every single chapter at the end. So while teachers are, like myself, are more than familiar with the content, it's really the approach that the NCCA are expecting of teachers in the classroom, including myself, that really there is a very, very different approach.
If I can come in there as well, Yvonne, just on that.
Humphrey. Yeah.
I think as we talked about already, the course is very broad. It's still a very broad course, and there'll be challenges in terms of getting through the course. And already I've heard Ija talking a little bit about teachers utilizing the flipped classroom approach, whereby students would study text and then they would come into class and they would do tasks in relation to that. And flipped classroom can be very useful for accelerating through a course to a degree. And I think the way we've structured our chapters will really help students and teachers kind of incorporate that flipped classroom approach into their lessons. Because what we can do is we can say, right, okay, we finished section 1. I want you to read through section 2 for homework tonight. You come back in the following day and you have a task that the students can do to check for their understanding built into the learning portfolio straight away. So, you know, we don't have to give them a task and then bring them back and design that task. It's already built into the book as well. So I think that can be something. That's probably an approach we'll probably try use more and more of. I think over the course of the next 2 years as we introduce the new course, to accelerate through and make sure that we're covering all aspects of the learning outcomes. But I think it can make maybe make our classes more interactive, because we're dealing with the assessment part rather than the actual going through the text part.
Thank you both for that. And actually, Humphrey, there were a couple of questions on the breadth of the new course, actually. So you've addressed it there in some of your answer there. So thank you for that. I'm sorry, it seemed to be all women who put the questions in this evening. Maybe if men don't ask for directions, they also don't ask questions. So I'm sorry, this is just the way it's rolling. So Joy asked, will there be plenty of secondary data examples for students to use and analyse?
Oh, absolutely. I think that was something that myself and Humphrey were very, very conscious of from the outset. You know, we were looking at the ag science syllabus, and as I've said, we have included an analysis questions in every single chapter, so the students will have plenty of opportunity to interrogate and interpret data, both from in the textbook and also in their learning portfolio. And something else just to mention is on not just data, we have lots of comprehension style questions as well for students to read, to really develop those kind of scientific skills as a scientific literacy, you know, which will lead them as a citizen in the future, really help them.
Thank you, Yvonne. Thank you. So then we have Breeding asked, are there any exam style questions included? Now, I think you've probably referred to that, but again, you might have more to say on that, Yvonne.
Yeah, well, I know we're all eagerly awaiting the release of the sample exam papers, as are myself and Humphrey, but just as we've unpacked all the learning outcomes, we're also going to unpack the exam papers as well, the sample papers. So when we do receive those, we're going to split those up for inclusion in our learning portfolio. So what I mean is, if you have a question on respiration, we'll put that into the section of the learning portfolio on respiration. We're not just going to stick all the sample papers as a unit at the back of the learning portfolio. We're going to tailor it, so we'll actually unpack all of that for you. And that means that we're holding back on the release of the learning portfolio until we have all that completed, because myself and Humphrey felt that would be a lot more useful to teachers to have it organised in that way, and will help the students as well with their learning. And throughout the book, in our end of chapter questions and in our learning portfolio, we've taken our experience from junior cycle science and also from Leaving Cert Ag Science and adopted the exam style questions that have been used in both those courses.
Brilliant. And it will be important to clarify that even though we are holding back publication of the full learning portfolio till we have those exam questions, when you get the textbook, there will be a sample of the learning portfolio and investigative logbook in there, so at least you can get a feel for that and see what that is like. As indeed, there'll be a sample of the teacher's resource book as well.
Just very quickly, if I can pop in for...
Sorry, absolutely, yeah.
Just maybe to make people aware that we're expecting the publication of those sample exam papers, 2 at higher level and 2 at ordinary level, to be early enough in April, maybe in the second week of April. So keep a little lookout for that as well.
Perfect. Okay. So we're approaching 8 o'clock, so I am thinking that we might wrap it up there. There are a lot of questions that we weren't able to get to this evening, but as I said to you, they will not go unanswered. We will get answers back to all of you. Just let you know that obviously the school book grant this year is covering Leaving Cert as well, so please do get in touch with your rep. If you are planning to adopt Evolution, they will make sure that you get the best value out of your grant. So please do get in touch with your rep if you're planning to adopt it. And obviously, if you adopt the book, you will get access to all of the resources on Gill Explore. You know, come the start of the next school year, everything will be up there on Gill Explore, ebooks, PowerPoints, quizzes, all of that wonderful stuff that was spoken about this evening, and the PowerPoints. And obviously, again, I think Humphrey and Yvonne both referred to it, that the items in there that need to be editable for you guys to tailor them to your own needs will be editable. So, you know, you have that as well. So an email will go out to you tomorrow where you will get a recording of this webinar. You'll get a link to the website where you can see that sample unit and the other sample digital resources of the program. That should be in the email as well. And a link to the website where you can connect with your local rep. Another thing then, just when we finish this evening, a survey is just going to pop up. There's literally only about 4 questions, I think, in it. A very, very short survey. We'd really be delighted if you could fill in, that in for us this evening. As a thank you, we're going to pick 3 completed ones out of a giant digital hat, and you'll get a €1 for all, a €50 1 for all voucher each for those. So it would be great if you could fill in that survey for us. So I think it just is left for me to say again, thank you very much to Humphrey and to Yvonne for this evening, for a great presentation and great answers to questions. Thank you both, and good night to all of you. Thank you for joining us, and enjoy the rest of your evening. Thank you.
Thank you all very much. Have a lovely evening.
Thanks very much. Thank you.